It's my pleasure to introduce our next guest from Andelyn Biosciences, Senior Director of Business Development and Strategic Partnerships, Brian Duber, and Head of Process Development, Plasma and Viral Vector Core Facilities, Andrew Marrero. As a reminder to attendees, if you'd like to ask a question of our speakers, please type it into the question box under the video player, and we will try to ask them on your behalf. The format for this presentation is a fireside chat. We'll go about 25 minutes, and we'll try to answer questions at the end. So to start, Brian, let me ask you to provide a couple minutes of introductory comments about Andelyn for investors new to the story.
Yeah, certainly, and thanks for having us here. So my name is Brian Duber. I'm Senior Director for Business Development and Strategic Partnerships here at Andelyn. Andelyn's CDMO that was spun out of Nationwide Children's back in 2020, but the CDMO has a lot of organizational experience that goes all the way back to 2006 as a viral vector core for Nationwide Children's. I wanted to give a little bit of a background on why we spun out, and I think that talks a little bit more to our story than a typical CDMO. So we spun out of Nationwide Children's because for the last 15 plus years, we were manufacturing preclinical and early stage clinical trial material, but we saw a lot of those organizations have to eventually leave the CDMO or the viral vector core to go to another facility to help them with late stage. So what Andelyn decided to do when we spun out of Nationwide Children's was to allow us to stay with those organizations. We wanted to have that commercial expertise, that commercial capacity, so that we can go from a client from pre-INT to post-BLA. So as part of that vision and that mission, Andelyn decided to name ourselves part of Andrew, who was 8 years old, that received the first Duchenne's muscular dystrophy program back in 2006, and Evelyn, who received the SMA treatment back in 2015. So as part of that, we wanted to focus specifically on AAV gene therapy and other viral vectors as well, primarily because we saw that there was a huge capacity constraint within the industry. A lot of that capacity is not necessarily capable capacity. We wanted to be able to bring our industry experience to these companies that are developing these diseases for rare disease, but also some more prevalent diseases more recently. And we can dive down into our expertise a little bit later as Andy kind of touches on our platform processes to how we can deliver to these patients.
Well, great. Let me switch it over to you, Andrew, and talk about where you're at in the status of your build out.
Sure. Thanks for having us. So we're located in the greater Columbus area, Columbus, Ohio. We currently have 3 facilities that are all located about, all of them were within a 10 or 15 minute drive from each other. There's the Andelyn Development Center. So that's where I'm sitting now. That houses all of our preclinical manufacturing and development activities, from analytical development to everything going all the way up through IND enabling tox study manufacturing. So it's about 20,000 square feet. There's 20,000 feet built out. There's a 40,000 square foot facility in total, with isolation suites for other viral vectors, whether for lenti, baculo, et cetera. Different platform processes that we're trying to develop, as well as analytical development and in shared common space. We took possession of that facility about a year ago in April of 2022. Then our Andelyn Corporate Center, this is our GMP manufacturing facility. So this handles all manufacturing from early phase clinical all the way through PPQ and commercial. It's a 180,000 square foot facility. It consists of 10 manufacturing suites, 8 upstream, 2 downstream, as well as dedicated fill finish support spaces. It's built completely unidirectional flow, single pass air for every suite. and dedicated air handlers for every suite, as well as warehousing, et cetera. It's when we endeavored to build that, design and build it, we really did it with multiple products in mind, in multiple different vectors in mind. Whether so that's hence the single pass air and in unidirectional flow. So it wasn't the cheapest way to build a facility, but it certainly was the most defendable and the most flexible. So there's also space dedicated to expansion, so we can double the square footage of that facility in fairly short order. All of the utilities are already in place, and there's a second floor build out that's planned as well that will add additional manufacturing space. And then down at the bottom is the Andelyn plasmid facility. So this was formerly the viral vector core and GMP manufacturing facility, and we retooled that with ADC and ACC coming online. We retooled that for plasmid manufacturing. We did this to secure supply chain and shorten timelines for manufacturing, as well as control quality. And that came online last November. The corporate center, we just got our full certificate of occupancy a few weeks ago, but we have actually been manufacturing in that facility since last August with a temporary certificate. So that's where we're at today, and with all three facilities at full bore now.
So, if you're able to now offer customers kind of an end-to-end platform approach, can you highlight more specifically what you would view as some of your strengths?
Sure. So when we built this platform, or started building this platform, actually, 15 years ago, we quickly realized even then that we had to build it for multiple products. It couldn't be too rigid. So there's a lot of things a platform can offer in terms of our clients with supply chain, consistency of material, speed of development, as well as consistency of operation and control. And one of the things we have always done is we made sure we were using the same platform from to support all the preclinical work all the way through phase 1-2, IND enabling phase 1-2, and now going into commercial manufacturing. So that allows for consistency in manufacturing, consistency in product, that really streamlines filings and smooths out the road when it goes into filing and PPQ. So there's lots of things it offers, but like I was saying, it doesn't do any good to offer those things if you don't have the flexibility to be able to support multiple products, multiple serotypes. So when we built it, we did it sort of in a modular fashion, liken it to building Legos, where each orthogonal step can be swapped out for another step, depending on scale, whether you want to be in suspension, or an adherent system. The production is fairly agnostic to serotype and transgene. The downstream purification, there's multiple different pathways that we can offer in that's all within the platform. And most importantly, it shortens development cycle. We understand PPQ, as well as the CQ, the critical process parameters to go into each step. So we can help guide our clients along and really make those development cycles for optimization and range finding, et cetera, go much faster and much more smooth.
So, the organization has a lot of experience with companies at the earlier stage. A lot of our investors have interest or investments in companies at that earlier stage. So I think it would be helpful to hear from you, what are some of the common mistakes that early stage biotech make that you persuade them not to?
Sure. Yeah, that's a good question. It's and every situation is unique. Some biotechs inherit certain things from licensures and things, and you have to roll with the hand you're dealt. But really, there's I think I've already said the word smooth like 3 times, but one thing that's always certain is smooth is fast. And it's very easy, in the current climate and as competitive as things are, to try and go fast. And all clients, all biotechs want to move quickly, but smooth is always quick. So don't go too fast. Don't ignore certain things, like platform, like development. Make sure you're in an optimized state, or at least a partially optimized state. Make sure that your raw materials are in an optimized state. The last thing you want to do is get all the way through phase 1, 2, and realize you have to redesign a plasmid because of some intronic regulatory element that the FDA is not too pleased about, or another regulatory body, or you realize that the platform you're in can't sustain the types of yields that's needed to support commercial manufacturing. So these are things to think of up front, and we're always happy to lend our expertise and our experiences and that's why we, whenever we bring a client online, we try to have these conversations as early as possible with them.
So maybe the other side of that question is, given that tech transfer and customer integration is critical to your success, based on your customer experiences, what have you learned to do better?
Well, you're speaking with, you mentioned tech transfer. That's something else we're fully capable of doing. But I would say it's really that client relationship. It goes back to our DNA in academics and collaboration and in working from the very beginning with our clients and our partners. We look at all of our clients as partners in those early on development cycles, or working through tech transfer. There's inevitably going to be development that's going to happen, even with a full tech transfer, or some level of optimization. So, get a good relationship with your CDMO, and CDMOs get good relationships with your client partners, and that's really that fosters the right level of communication, the right level of conversation, and gives you the highest probability of success at the end of the day.
So, Brian, I guess another moving target is just the current market environment. 2 years ago, the conversation was all about how there was not enough capacity to meet demand. Fast forward to today with access to capital much more expensive. We see the early stage companies pulling back to extend their cash. Tactically, as you're still in a bit of a build-out mode, how have you adjusted your corporate strategy?
So like I had mentioned before, we spun out of Nationwide Children's to be able to be that end-to-end service provider to a number of biotechs, and people from folks that are working with foundations all the way up to folks that are late stage. So what we've done to help facilitate that is build out or do a stage build out of our facility to ensure that we are able to deliver on what the clients need today. So that's that capable capacity to where we have platform processes already in place, so we can help expedite the client's pathway to and through the clinic that way. While we also do retain a significant portion of both facilities as growth areas. So being able to pivot and work with the market, whether or not it's different viral vectors, or continuing on the same thing that we're currently doing. So that could be expanding on plasmids or expanding the amount of GMP suites. It's all very, very doable for us right now in our current facility.
Okay, so Andrew, let's dig a little deeper into some of your proprietary technologies. Starting with your AAV designed components. Can you describe any key advantages of your HEC 293 suspension cell line, along with your cap rep and helper plasmids or your capsid designs?
Mm-hmm. So, yeah, starting with our cell line, what we really tried to do was bring the entire supply chain under one roof, from raw material all the way through analytics and disposition. And the cell line's a key component of that. Several advantages to having our own. It's we're able to develop a high performing cell line, that can package well across multiple serotypes and transgenes. So it has high packaging efficiencies, high performance rates, high purity. Also, we can guarantee clonal isolation of that cell line and licensure. So we really tried to make that cell line accessible to our clients. And so, some of the other more commercially available cell lines have some fairly extensive licensure restrictions. Ours is, very favorable. So these are that's the cell line. When it comes to the other critical raw material, the plasmids, like we said, we brought that manufacturing in-house. We also brought some of the plasmid design in-house. We've learned a lot of lessons over the years, seeing different constructs, different construct designs and philosophies behind that, regulatory issues. So we're really able to lend that longstanding expertise to that, as well as, and we've developed some Gen 3, we call them capsid backbones and vector backbones, so that to eliminate RCAV, to eliminate other non-desirable regulatory elements, and make sure that the plasmids themselves are manufacturable. And then when we apply those in our transient transfection process, they produce a high quality, it's a high quality and high yielding product at the end of the day.
Okay, and can you describe some of the advancements you're achieving in lentivirus production?
So this is a new endeavor for us. We're developing that platform now. We're using the same guiding philosophy that you see here. It's and the same with the same types of orthogonal steps in both suspension and adherent. We'll have that as an offering for preclinical manufacturing. We're targeting the end of the year for that.
Okay, and switching to the downstream side, what are you doing to improve the performance of your assays to characterize potency, purity, and other important product attributes?
So this is something that's been, evolving quite a bit as well. And is always, top of the list for regulatory issues in all the regulatory bodies. Doing a lot of things to increase, precision and accuracy, migrating from, legacy qPCR, which, had very, very low levels of precision, to ddPCR that have very high levels, moving from semi-quantitative, or more qualitative assays to fully quantitative assays, like, SDS page, into quantitatable assays for purity. A lot of things like that. A lot of life cycle improvements as well.
Okay, and in terms of fill finish, how can you turn this into a competitive advantage?
In terms of fill finish, so we have fill finish capability that's semi-automated. We're also going to bring online a fully automated high volume fill finish fill finish system. But it really, like I was saying before, really it integrates the entire life cycle of the product, right? From concept all the way through, and then as well as the manufacturing life cycle as well. So, we can control the supply chain completely vertically integrated from raw material all the way through fill finish and disposition.
Okay. We've heard a lot of talk about AI and its potential. Where do you see implementation of AI-specific apps could improve the efficiency of your current manufacturing processes in the future? And again, I'm not talking about, like a dedicated robotic arm or something, but
yeah. So I think this is something it's all the buzz, and it's something that will definitely impact, the gene therapy field from a clinical side and a manufacturing side as well. I can see, AI seems to work as well as the information that you put in it. So it's going to be really interesting to see how it can become. I can see it becoming sort of a predictive model. It can also, predict efficiencies, like you're saying. We have 15 plus years of data. This is something that I think Andelyn is unique in, where we've been manufacturing clinical grade vectors for, since 2000. I think we first released was in 2010. So, we can compile that wealth of data. You multiply that with AI's capability of using that data for predictive and predict and learn from it. And then it's really going to be interesting to see where it goes. I have no, hard and fast predictions of where it's going to go at this point, but it's going to be interesting to see.
Okay. As we're now moving past the urgency of addressing the COVID-19 pandemic, how is this changing, access to the FDA or, loosening constraints on your supply chain?
So in terms of the FDA, now that we're coming out of the pandemic, we're actually able to interact with them and have meetings. This was something that was difficult to do during that period of time. So hopefully we'll see a lot more filings coming down the road and INDs as a result. But so that's one thing. I know we've had some recent meetings and things that have been very positive, faster turnarounds and things that it's going in the right direction again. For the rest of the medical, biomedical industry, and in terms of supply chain, there's still impact, right? But this is where relationships come into play in a big way. We were able to stay whole through the entire pandemic, and we never lost manufacturing capacity or capability. And that comes from a lot of proactive supply chain maneuverings, and our head of supply chain, John Rush, he's been fantastic at staying ahead of things. But it's really, it just shows that building those relationships is critical, and I think it's going to continue to be that way for the foreseeable future.
Okay, well, Brian, let me ask you one final question, and kind of serve as a way to summarize all the things we've just discussed. But how is Andelyn differentiating itself as a preferred CDMO partner?
Yeah, so I think it kind of ties into a lot of the questions that we touched in today. So from a supply chain standpoint, we have a minority investment from Danaher, Paul, Cytiva. So maintaining those relationships and utilizing those connections to be able to have a very secure supply chain, as well as continuing our relationships with Corning for those particular materials, and then having an eye on what we need as far as GMP materials and other critical raw materials is very, very important for our clients. Additionally, I think it's a little bit different than most other CDMOs. We're not necessarily only focused on full process development and GMP programs. We interact very well and with early stage clients. So folks that are in foundations or even PIs at the university or a research institute level, being able to plug into them and act as an extension of their lab is something that we do very well. Making sure that we're enabling their team to be able to produce something that is of the top quality very quickly and in sufficient amounts, so that at the end of the day, we're getting these life-saving treatments to patients where there's a significant unmet need.
Well, great. Well, we've reached the end of our time here, but I want to thank you both for joining us today.
Thanks for having us.
Yeah, you're most welcome. Thanks for having us.